Assalamu alaikum. We are going to talk about epigenetics and development. And we are going to specifically cover a topic which is referred to as epigenetic cell memory. It was a phenomenon which was discovered in fruit flies, and the proteins which are involved in this phenomenon of epigenetic cell memory and development, they were best characterized in Drosophila. And eventually, what we have learned over the last 20 years that these groups of proteins which contribute to maintenance of epigenetic cell memory, they are actually conserved across evolution in invertebrates, in the vertebrates, and that is the best described phenomenon of epigenetics in development. So let's learn what is epigenetic cell memory and how epigenetics plays a role during our development. So we learned during earlier part of our lectures that in fruit flies, in Drosophila, maternal genes, they set up the body axis, and these establishment of anterior, posterior, dorsal, ventral body axis, it actually is due to specific gene expression patterns which are established due to action of maternal genes. And maternal genes, they lead to activation of gap genes, which are transient, just like maternal genes. Gap genes, these are transcription factors, they activate, then they act on pair rule genes, which are also transcription factors like, even-skipped and Fushi-Tarazu. And these pair rule genes, they act on segmentation genes. Segmentation genes are basically, groups of genes which contain cell signaling proteins, transcription factors. It's a diverse class of genes. And these segmentation genes, they eventually act on or activate Hox genes or homeotic genes. And homeotic genes are the ones which give special identity or specification of different cell identities in fruit flies. So if I simplify this whole thing, I would say that development, it starts from single cell, which is the zygote, which results from process of fertilization, the one end of sperm cell and the one end oocyte, which makes the diploid zygote. Now, this cell, it undergoes cell division into two. This further goes and keeps dividing. During this development, the earliest distinction between different cell types or between the cells within the embryo, it takes place at the stage of gastrulation, where cells are eventually, clustered, or they become the ectodermal, mesodermal, and endodermal cells. This is the stage, the gastrulation stage, where cells, they can be identified at molecular level, at gene level, that there are three clusters of cells in terms of gene activity they are having. Now, during this development, there comes a stage where you'll have, for example, we just focus on, out of hundreds of cells there, let's focus on just 4 cells. These cells, their fate gets determined when cell is growing like this, embryo is growing like this, cell fates gets established. We call this stage cell fate determination. For example, this cell is going to make future eye. This cell is going to make future heart, kidney, liver. In a multicellular organism we are talking about. Okay? Generally in multicellular organism we are talking about. Now, the cell shape, it may not be looking different. They all may look same. But this is, during development, a very important milestone has arrived, which means cells, they are committing to different fates. Now, we know these cells keep dividing. Okay? They keep dividing mitotically. Like this, mitosis is taking place. So the fate of eye cell is being inherited to daughter cells. They divide into two, then further 2 cells, so through mitosis, eventually the cells which are going to constitute eye we eventually see here differentiation. So here what we saw, cell fate determination. So different cells commit to specific cell fates, and eventually, let's say here we see liver. Here we see kidney. So the fate of cells, which was established long ago during early development, this is maintained through successive cell division, through mitosis, and eventually we see differentiation. Now, when differentiation takes place, now you can clearly see, the cell shape and, morphology clearly distinguishable from the early cell type, where the cell fate was determined. Now, we call this cell memory, going from establishment of cell fate all the way to differentiation. This is called cell memory because different cells, they are going to remember their original fate. All these cells, through successive rounds of mitosis, eye cells, every mitotic cell division is going to remember, the daughter cells are going to remember that they are committed to eye lineage, or these cells are making the kidney lineage or heart cell lineage. So when each daughter cell remembers the original fate which was determined early on during our development, we call cell memory. And why it is called epigenetics? Because, for example, we are talking, let's take our own example, the humans, the multicellular organism. This oocyte contains, the zygote contains 46 chromosomes. Each subsequent cell here, all the somatic cells, they contain 46 chromosomes now because we are growing through mitosis. But when the cell fate determination takes place, they start expressing different genes. The establishment of cell fate, it is due to expression of key developmental genes which are either switched on or switched off. Some of them will lead to eye development, the others heart, kidney, liver. So this, a very important concept in molecular biology is differential gene expression. So this, although each cell, each somatic cell, it contains same set of 46 chromosomes, same genes present on those chromosomes, but they are attaining different fate. That is due to the differential gene expression. For example, I draw it here again. These arrows represent, the mitotic divisions, successive mitotic divisions. This is same number of chromosomes, 46. Here, cell fate determination took place. Here, eventually, differentiation took place. And, memorizing all this phenomenon is called cell memory. But when cell fate gets determined, different cells, they start expressing different genes, although they all contain same 46 chromosomes, which means same set of genes. I further simplify this. For example, here you have eye fate, liver, kidney, heart, muscles, etc. And just write down different genes. Different key developmental genes, A to Z. These genes, they are all chromosome. Each cell contains same chromosome. There is no distinction between cells in the form of presence or absence of chromosomes or genes, because they all contain same pair of chromosomes. Now, however, what happens at the time of cell fate determination, due to developmental signals, the cell signaling, what happens, for example, A becomes on in eye, B is off, it's on in liver, off in kidney, off in heart, muscle. C becomes on in eye, off in liver, D is off in eye, on in liver, E is only present in heart, F is present only in the kidney, and so on. Now you see, gene A is present in every cell, but it's not being transcribed, or its gene product, which is a protein, it is absent in liver, heart, kidney. This is how, when you will go from A to Z, and if you analyze I specific gene expression pattern, you will find A is there, C is there, M and O is there, Z is absent, etc. So you get what we call differential gene expression of I, or what I call cell type specific gene expression pattern of eye, or cell type specific gene expression pattern of liver. This gets established at the time of cell fate determination. This eye specific or cell type specific gene expression of eye cells at the time of cell fate, they lay down the foundations of eye cell fate, which is inherited through successive mitotic divisions. Each daughter cell remembers the eye specific or characteristic eye specific gene expression pattern all the way up to differentiation and all the way up to my death. Okay, so this differential gene expression pattern, which gets established at the time of cell fate determination, there are two things. One is establishment. Very little we know about the establishment phase, because there is massive cell signaling going on during early development. Here we don't know the uterine environment within the womb of our mother. So this is one of the key challenges, a $64 million question in epigenetics, how different cell fates gets established. But the maintenance part, and then the second part is maintenance of these differential gene expression patterns which contribute to cell fate. So one is establishment of cell fate, the second is maintenance, which we refer to epigenetic cell memory. Why we call this epigenetic? Because as we learn and we know that the DNA sequences, the genome, the chromosomes, etc. is same in all the cells, but their regulation at the epigenetic level is different. For example, this gene in eyes is on, is being transcribed. Here it's off, and it may be due to DNA methylation or histone methylation, whatever epigenetic modifications we just talked about in the previous lecture. So once these epigenetic marks, for example DNA methylation, is laid, we just learned in previous section that these epigenetic modifications, for example DNA methylation, it can inherit through mitosis, through cell cycle. DNA methylation is known to be one of the earliest cell memory mark. It was named as cell memory mark. So the end result is, although you have same number of chromosomes, same set of genes, but you are having different fates because you are selectively switching on and switching off genes, which we refer to as differential gene expression, through epigenetic phenomenon. So DNA sequences of any given gene, for example gene A, it's 100% same in eye, liver, kidney, heart, muscle cells, etc. But due to epigenetic phenomenon, it's off here in liver and kidney and remains on in eye and kidney, for example. So this answer to this question was well described or well characterized in fruit flies. And what we learned, that I just give you a recap of what I told you earlier, that in fruit flies, due to maternal genes which are deposited by mother at the time of oocyte formation, maternal genes, and you may recall we had bicoid, nanos, caudal, hunchback, etc. So for example, bicoid was giving you expression on the anterior side. When fertilization takes place, the RNAs which are deposited by mother are translated, and on the posterior side you have the nanos and caudal. Okay. They are transient. They activate the next group of genes, which are, for example, Krupel, NEPs, etc. The next class is called the GAT genes, because if there are mutants in these genes, you see much bigger gaps in these embryos. For example, Krupel is expressed in a big region like this. These are, as I said, these are transient as well. They activate the next class of genes, which are called pair rule genes. And this expression of pair rule genes, it coincides with the signs of segmentation. So, for example, we talked about, the E1 skate and Fushitarazu. They coincide with, the onset of segmentations, and eventually they lead to now the activation of the pair rule genes. They activate the segmentation genes. And what we see at the time of segmentation, we clearly see, the formation of the head region, the thoracic segment, the head region, and abdominal segments. All this is at the embryonic stage. And what we also learned, that by the time you have pair rule genes, the embryo is syncytial, but as soon as the segmentation genes come in, cells are cellularized. They are diverse class of genes. They are cell signaling proteins, etc. Now, what is next? Because you cannot just go on like this. So in fruit flies, after segmentation genes, what you have, so these segmentation genes, they lead to activation of genes which are called homeotic genes. And these homeotic genes, they are expressed in very specific clusters, in specific regions. And I would like you to go back to our earlier lecture where we talked about homeotic genes. There are two clusters in Drosophila. One is called bithorax complex, the other is called antennapedia complex. So in fly up to, parasegment, if I remember correctly, it's parasegment 4. Up to parasegment 4, we have, expression of antennapedia complex genes, and all the way from 5 to 14, we have bithorax complex expression. And you may recall we had, from 5 like this, in a slight overlapping regions, we had here red one is UBX, ABDA and ABBB. You recall all these lectures. Now, what happens? These homeotic genes, they are not transient. They are the selector genes. They are the genes which now seal or specializes cells. For example, UBX, absence of UBX means, wing imaginal disk or wing fate. Cells specialized which are going to make wings, they are not going to express UBX, and the presence of UBX, which is a homeotic gene in the bithorax complex, is going to make Halter. ABDA, ABDP, ABDB is expressed in the last posterior most segments in the abdomen, and ABDA in the anterior abdominal segments. Now, we said these are transient. So basically, the cell fates, they are determined already here. Already, cell fates are determined by this stage. You can see 14 different segments. So the foundations for cell fate determination is laid down here. It eventually stops at the selector genes, the bithorax complex and antennapedia genes. The big question is, now we cannot pass on the baton to next class of genes. So these genes which gives identity to specific cell fates, which seals, which stamps the cell fates, their expression is maintained. And their expression is maintained by two groups of genes. They are called Polycomb group, and the other is called Trithorax group. Polycomb group are silencers. For example, Polycomb group genes or proteins, they are going to maintain developmental genes, for example, UBX in the off state, switch it off in the wing cells. And Trithorax are antagonists of Polycomb. They are the activators. They maintain genes in active state. So UBX will be maintained in the on state in Halter by Trithorax group, but UBX is maintained in the off state in the wing imaginal disk by Polycomb. So you can think of, a seesaw kind of thing, where if Trithorax group overrules, gene is going to be on, and if Polycomb group overrules the Trithorax effect, gene is going to be off. Now, both of them, they are chromatin binding factors. Both of them contain multi-protein complexes, and they are epigenetic factors. In the next section, I'm going to talk about the detailed function of Polycomb and Trithorax, how they play a role in epigenetic cell memory, how they ensure a specific gene remains off by Polycomb, or how Trithorax ensures gene remains on. In the epigenetic cell memory, remember, cell fates established. Now these cell fates are going to be maintained through the phenomenon of epigenetic cell memory, and Polycomb and Trithorax are two major gatekeepers or players which govern the phenomenon of epigenetic cell memory. So stay tuned. In the next lecture, we are going to see that.